Hi, this is Andy. Welcome back to Med School EU. And in today's video, we're going to continue to talk about reproduction and inheritance. And more specifically, today we're going to take a look at classical genetics, which entails chromosomal theory of inheritance, as well as inheritance patterns. And as I've mentioned in previous video, we're going to take a look at Mendel's genetics and its modifications in the present day genetics. So first, let's discuss the chromosomal theory of inheritance, and this one should be pretty obvious to us by now, looking from Mendel's genetics, because Mendel pretty much discovered all of the same things with his garden peas experiment. And basically what this theory says is that it was first of all composed by a scientist named Sutton. And he concluded that genes and their alleles are carried on the chromosomes. And that is basically just the chromosomal theory of inheritance. So he deduced that genes and the alleles are carried on chromosomes. So these are obviously homologous chromosomes, and they're going to have a gene right here. So that's the gene. We can label this as gene. And each one of these yellow markers is going to be a separate allele. So let's say it's for allele named A. So A allele. And this is going to be the dominant one, of course, with the uppercase. And lowercase a allele. So alleles are just versions of genes. We did the definition last time. And the theory simply states that alleles and genes are found on chromosomes. So now we're going to take a look at modifications to Mendel's hypotheses, because Mendel discovered a lot of amazing things, and with his 3 hypotheses, they were great for the time being. However, his hypotheses do not describe genetics completely. And in order to describe how our genes and alleles actually work, there needs to be some modifications to the 3 hypotheses he has made, because not all genes are simply dominant and recessive over the other. So we're going to take a look at some of these modifications. And the first one is going to be incomplete dominance. And the best example for humans to take a look at the human genome of incomplete dominance would be the sickle cell disease. Because in sickle cell disease, even when you're heterozygous, you are going to have the sickle cell trait. So you're going to exhibit some sickle cell symptoms even though you are heterozygous. So you have a dominant allele present, however, it's not going to completely mask it. So let's break this down with a simpler example. So if we have an individual who is medium height, so for example, he has the T allele and the little t allele, and we're going to crossbreed with another medium height individual. So they're going to produce the big T, big T, big T, little t, big T, little t, and 2 small t's. So if we take a look at the genotypes, we're going to end up with 1 tall individual. We're going to have 2 of these heterozygous, and we're going to have a homozygous recessive. And the phenotypes that they would produce is going to be tall. We have medium height, and we have small. So here, if we were to simply go ahead with Mendel's hypothesis, and we would just assume that the dominant allele is always going to be completely dominant over the recessive, then this one would not be medium, it would just be tall. However, of course, that's not the case, and most of our alleles and most of our genes do not work in this dominant recessive fashion. They typically work with incomplete dominance. And this is just a direct example. And the most common example that we have in our human bodies is the sickle cell disease. So sickle cell disease, same thing. If you are heterozygous for sickle cell, let's say it's H and H, you're heterozygous, you're going to experience something called sickle cell trait. Sickle cell trait. So you're not going to have the sickle cell disease, because sickle cell disease is going to be recessive, homozygous recessive. So this is going to be homozygous recessive, and of course this is homozygous dominant right here. However, if you are heterozygous, so you still have the allele that is going to produce sickle cell red blood cells. It's going to produce these terrible blood cells that cannot carry oxygen very well. However, at the same time, you're also going to produce a sufficient amount of healthy red blood cells. So you're still going to experience some symptoms that will not be as terrible as having the complete homozygous recessive gene. And this is another example of incomplete dominance, because it's in heterozygous, according to Mendel, you would have simply just had the regular red blood cells and you wouldn't even notice. Nobody would even know that you have the recessive allele. However, it doesn't work like that in real life, because for sickle cell disease, we have a sickle cell trait, because this recessive allele is still going to be expressed to some extent. And remember how I said previously that the dominant alleles, they do not directly mask over, or they do not directly inhibit the production of the recessive allele protein, meaning that both alleles are going to produce their proteins, their designated proteins, and it's simply because the dominant allele is going to be able to produce them more, or they're going to be expressed more. However, one main component you should know is that both proteins, both genes, are typically expressed in a heterozygote. And just the fact that one is dominant over the other does not constitute that it's being inhibited. And now we're going to take a look at the other two modifications, which are codominance and multiple alleles. And we can explain this with one simple example that all humans contain, and that's our blood cells. So our blood types, of course, we're going to have the A. So let's label all of these. We have our A blood type, we have the B blood type, we have the AB blood type. So everybody is either A plus, A negative, B plus, or B negative, AB, and we have the O blood type. And each of these blood types, they have antigens, meaning that they have these receptor molecules on their red blood cells and their blood cells that the immune system is not going to target. So we could label these as the blood type A. So the A will be the antigen for blood type A. B will be the antigen for blood type B. Then AB will have both A and B, and O will have none. So we'll have nothing there. And so what this really means is that the immune system is never going to attack the A antigen for blood type A. Immune system will not attack B antigen for blood type B, because these will be recognized as self cells. These are the cells that in our own body. So the immune system will know that it's its own body, and it will not attack these blood type antigens. However, each blood type here is going to have antibodies. So you're going to have antibodies floating around in the blood that will be able to trigger an immune response if they're tagged by a different kind of blood type. So here we have the antibodies for B. B have antibodies for A. AB blood type do not have any antibodies. And the O blood type have both A and B. So here, if we go over the accepted transfusions, we could say that for blood type A, you could do a transfusion with another blood type A or blood type O. And if you're looking at B, it's going to be B and blood type O. And for AB, it could be any. So A, B, AB, or O. So this is the universal acceptor. AB blood type is universal acceptor, and O is the universal donor, because for O, the only accepted blood transfusion is going to be blood type O. Now, how does this all relate to Mendel's modifications of codominance and multiple alleles? Well, the blood types are all coded by one gene, and this one gene is I. This is the dominant version of it, and this is the recessive version of the gene. So here we're going to describe, because there's going to be multiple alleles, as you can see here, A, B, and I, that exist for the single gene. So originally, Mendel mentioned that there's two versions of each gene. So there's two alleles for each gene. However, as this example states, we have three versions of the gene. We have the dominant version of A, we have another dominant version of B, and we have the recessive version of just the I. And of course, if we test cross with another one, just to see what kind of combinations we can get. And these would be kind of the frequencies that we would receive, and you will see from this test cross why we get so few O blood types and why they're so rare. So what we have is we're going to test cross, and I'm just going to point out immediately with the colors of which blood types we receive. So if we have here we're going to have I, uppercase I, A. So this is going to be dominant. Now, if we're looking at here, this one, I, A, and the recessive. So this is still going to be blood type A. And of course here we got IA and the recessive. So this is all, these three are going to be the genotypes for blood type A. Now if we're looking at blood type B, we would have this one right here. So these would be the blood type B, and as you can see here, the I is recessive, meaning that blood type B is dominant, and that's the blood type B that's going to exist, even though that is heterozygous. And the next one we can have is going to be this codominant one. I'll label it in white. It's going to be blood type, so here A mixed with B, and that's this one right here that we get, AB blood type. It's the codominant, because both dominant alleles are mixed together and they're called AB. They're both expressed and they're both used in equal frequencies. So one is not masking over the other, one is not dominant over the other. They're both codominant. They're both expressed and they're both used in the bodies. Now, and same thing happens right here. And finally, for our recessive allele, we have blood type O. So this one right here is going to be blood type O. As you can see, all of this exhibits multiple alleles, and this one right here specifically, because A and B are codominant, they're going to be AB blood type. It's this whole separate blood type that we have. So the biggest likelihood of being blood type A or blood type B, and the least likelihood is being a blood type O, because it is recessive. So you have to be homozygous recessive in order to be blood type O. However, you can appreciate that now we know that Mendel's hypotheses were amazing. They were great for the start of genetics. However, now they have several modifications with incomplete dominance, as we've mentioned here, and a lot of the genes exhibit codominance as the ones in the blood type. So blood type A and blood type B are codominant alleles, and they also exhibit multiple alleles because our blood types, they have 3 different alleles: blood type with the IA, IB, and the little I. So it's important to keep in mind. However, although there are multiple alleles in the population, so in overall population, there's going to be multiple alleles. Some will be just the blood type O, some will be blood type B, some will be blood type A. However, for the individual, they're still only going to have 2 alleles existing in their bodies, in their cells, just the 2 alleles, as you can see for all of these. You cannot have 3 alleles on the homologous pair of chromosomes. Each individual is going to have just a pair of alleles, just like Gregor Mendel has mentioned in his hypotheses. However, for the population, there's going to be multiple alleles. This concludes our lecture on classical genetics. In the next lecture, we're going to start looking at things like replication and the structure of DNA and something called molecular genetics.